Now, I would like to introduce our webinar speaker, Dr. Anne Rowley. She is the Professor of Pediatrics and of Microbiology Immunology at the Northwestern University Feinberg School of Medicine and the Stanley Mann Research Institute, and an attending physician at the Division of Infectious Diseases at the Ann and Robert H. Lurie Children's Hospital of Chicago in Chicago, Illinois. She has devoted most of her 30-year academic career to the study of clinical aspects, pathology, immunology, and pathogenesis of Kawasaki disease, a serious and increasingly recognized worldwide pediatric problem. The title of her presentation is Cloning the Plasmablast Response to Unlock the Pathogenesis of an Inflammatory Disease of Unknown Cause. So once again, please use all the question pane for your questions. And again, we will share the slides and post-webinar file. And I'm handing over the cursor to you. Please take it away.
Okay, thank you, Karsten. And thank you all for joining today to listen to what I hope will be a useful conversation for you, not just about Kawasaki disease, but about potentially the study of other infectious and inflammatory conditions. I have a few disclosures, funding, and a patent application. What I'd like to do is to describe a little bit about Kawasaki disease. I suspect many of you on the call may already know about it, but just in case. And then I'd like to move to discussing the peripheral blood plasmablast response to infection, what that is like. And then I'm going to describe for you how we've prepared Kawasaki disease specific monoclonal antibodies from the plasmablast response to the illness. And we've worked to determine the targets of these antibodies. And then I will describe our progress towards developing the first ever, to my knowledge, Kawasaki disease serologic assay from these studies. So Kawasaki disease is a febrile illness of young children that is manifested by a variety of clinical signs and symptoms, which are depicted here, including rash, red eyes, red lips and tongue, red swollen hands and feet, swollen glands in the neck. All of these features actually resolve spontaneously, even if there's no treatment given. But the reason that it's an important condition is because children can develop coronary artery aneurysms and aneurysms occasionally in other blood vessels in the body, but most severely the coronary arteries. And this, in the most severe cases, can lead to thrombosis in these aneurysms with myocardial infarction in even young children. On rare occasions, these aneurysms can rupture and cause sudden death. And then later, the dilated arteries can actually develop stenosis with progressive obliteration of the lumen from a proliferating modified smooth muscle cell process that we don't really understand. So depicted here is a heart from a child who unfortunately died of Kawasaki disease, and the left coronary aneurysm and right coronary aneurysm you can see are very, very large and actually bigger than the aorta and pulmonary artery. And it's a cardiac catheterization picture demonstrating very, very large swollen coronary arteries. So the cause of this disease has really remained unknown for the more than 50 years that we have known about Kawasaki disease, from Dr. Kawasaki's initial description of the condition in Japan. But there is a number of features of the illness that really point towards this being an infectious disease. Some of these features I've listed on this slide, including well-described epidemics of illness in Japan, and there have been epidemics in other countries as well. And in all three of the Japanese epidemics, the epidemiologists did careful study of the cases and where they were located in the country, and in all of them, It started in one area of the country and spread in a geographic manner through the country over a 4 to 6 month period. They noted less spread when there was a high incidence area blocked by high mountain ranges, which suggested to the epidemiologists that people were needed for spread of this disease. And there's a higher attack rate in siblings, interestingly, with 2 peaks of onset compared to the index case, one peak on the same day as the index case, suggesting common exposure to some pathogen, and then another group at 7 days after the index case, suggesting that this might be the incubation period for this disorder. And here I've just shown you one of the pictures from the epidemiologists in Japan for one of the epidemics where the illness began in one part of the country and then spread geographically out from that initial starting point. Leading some of the most well-known epidemiologists on Kawasaki disease in Japan to state that the suspected agent must be one that generally exists in the community and spreads from community to community with high speed. There are many other features of Kawasaki disease that suggest that there may be a ubiquitous viral agent as the cause, including the young age group affected. We don't see much Kawasaki disease in the first couple months of life, suggesting that perhaps maternal antibody is protecting them. And then we see a peak of illness in children aged 6 months to 5 years. And then after that, we see occasional cases, but not as many. And then in adulthood, it's almost unheard of. And this strongly suggests that there's a ubiquitous agent that's causing this condition. As I mentioned, the clinical illness is actually self-limited, which would support a potential viral etiology. These patients do not respond to any antibiotic that's ever been tried. There's a very high prevalence of the condition in Japan, which strongly suggests that there are genetic factors that predispose to the development of Kawasaki disease. And right now, the numbers are approximately 1 in 65 children in Japan develop Kawasaki disease by age 5. So that is so prevalent, it does fit well with the idea of a ubiquitous infectious agent. In some of our other work, we've noted upregulation of interferon response genes in the lung and coronary arteries in Kawasaki disease. And we've also identified the presence of inclusion bodies with virus-like particles in close proximity in the bronchial epithelium of children who've died of this condition. And our other work has demonstrated that there's a prominent IgA immune response, suggesting maybe a mucosal portal of entry for the pathogen. And here's depicted some photos of the inclusion bodies that we've demonstrated in the bronchial epithelium. They're seen here in brown, using an immunohistochemical assay, and in green using an immunofluorescent assay. And when you look at these by electron microscopy, although we don't have glutaraldehyde fixed tissue to examine, what tissue that we have shows an inclusion with what appear to be virus-like particles in close proximity. Now, we detected these inclusions from antibodies, synthetic antibodies that we made in the past. We were able to identify clonally expanded immunoglobulin alpha heavy chains in coronary arteries of children who died of Kawasaki disease. At that time, the technology was such that we were not able to match up the correct light chain that would go with that heavy chain. And so our antibodies, although they identify these inclusions, we were not successful in using them to identify the specific antigen. So our hypothesis from our prior studies was that the KD agent is a persistent RNA virus with a respiratory portal of entry. I'm not going to go into detail the RNA and the persistent aspect, but this can be seen in our prior studies. And then in this study, we had a very basic sort of premise that Kawasaki disease patients are very likely to make antibodies to the causative agent because we all make antibodies to any infectious pathogen that we encounter, and that if we developed antibodies that had the correct light and heavy chain partners, that we should be able to identify the specific antigens that were involved in the process. Enter peripheral blood plasmablasts. There have been a lot of advances in molecular immunology in recent years, and it has been demonstrated that about 75% of peripheral blood plasmablasts at 1 to 2 weeks after infection are antigen specific. A lot of these studies derive from studies of dengue, influenza, and HIV as well. And it became clear that that shortly after infection, our peripheral blood plasmablast pool, which is the small pool, these are the precursor to plasma cells. They don't constitute a high percentage of our peripheral blood B cells, but and they're generally fairly clonally diverse. But right after we've encountered a pathogen, a couple weeks later, in the first week or two, they become very directed towards that infectious agent. And one investigator stated, when you examine peripheral blood plasmablasts, sort of an ideal one-time point diagnostic immune surrogate for serology during acute infection. So we decided that we would isolate single peripheral blood plasmablasts from KD children and characterize them, make the antibodies, and use the antibodies to identify their antigens. So basically, we take a small amount of blood from a child with Kawasaki disease. In our early studies, we did it up to 3 weeks after fever onset, but really for most, and that turned out to be a little bit too late. So 1 to 2 weeks is optimal. And then we isolate peripheral blood mononuclear cells and stain the cells. And the cells that we're looking for are CD19 positive, they're B cells. They're CD3 negative, they're not T cells. And they're highly CD38 and CD27 positive, which is the markers that show you that they are plasmablasts. We single cell sorted these plasmablasts into individual wells of 96 well plates, and then we performed RT-PCR to amplify the immunoglobulin heavy and light chain variable regions of the individual plasmablasts. And we did this on on over 1,000 plasmablasts from 11 Kawasaki patients. We identified the clonally related plasmablasts in each patient's plasmablast pool and some highly mutated IgA plasmablasts. And then we produced these in 293 cells. And because we had the peripheral blood plasmablasts in single wells, and there was 1 cell per well, we knew then what light chain was the appropriate partner for the heavy chain. We kind of skewed our data set towards children who had either coronary aneurysms at diagnosis or had a high risk of developing coronary aneurysms. Because Kawasaki disease is a clinical diagnosis, and because many of the features of Kawasaki disease, such as fever, rash, red eyes, are seen in a very large number of conditions in children, we decided that we needed to skew our data set so that we were very sure that the patients had Kawasaki disease, because this is a very distinctive feature of Kawasaki disease that is seen in very few other illnesses in childhood, if any. Some of these patients did not have all the clinical symptoms. 4 of them did not have all the features of Kawasaki disease, but 2 of these had coronary aneurysms, confirming the diagnosis. And we did this just over a little bit of more than a year, but encompassing multiple seasons of the year, since many have speculated that there might be different causes for Kawasaki disease by season. And then we wanted to have diverse ethnic groups that we were studying, and we had a variety of different ages and about equal girls and boys. All these patients received the treatment for Kawasaki disease, which is intravenous gamma globulin, and because they were high-risk Kawasaki disease patients, we also gave almost all of them, adjunctive prednisone. The blood was obtained in this time frame at a median of 14 days, and we sequenced about 100 plasmablasts per patient. So from these plasmablasts, we identified 42 sets of clonally related plasmablasts. I want to emphasize that each set was in an individual patient. So one patient would have multiple plasmablasts that were clonally related. Some of these had more than one isotype per set. In other words, it was the same plasmablast variable region sequence, but one was an IgM, one was an IgA, one was an IgG, strongly suggesting there was an immune response to an antigen occurring. And then, as I mentioned, we had some highly mutated IgA plasmablasts that we also identified, and we made about 60 monoclonal antibodies. The isotypes of the plasmablasts are what one would really mostly expect from peripheral blood, and we didn't see any particular heavy chain variable region skewing. So we had to decide what we were going to do with these 60 antibodies or 61 antibodies, and we needed to develop an initial screening method to decide which antibodies were highest priority for our future studies. What we chose to do, because our prior antibodies had identified these inclusion bodies, we said, well, if these are KD-specific antibodies, they also should identify the inclusion bodies. So we went and did immunohistochemistry to see whether each antibody was binding to the inclusion bodies that we had previously identified in KD bronchial epithelial tissue. And here's an example of strong binding of one of our antibodies to the inclusion bodies. We had 10 that strongly bound, and basically this is an arbitrary, we just could see them without having to use very high power. And then we had 22 that were more weakly binding, where we had to use higher power to observe them. We found in these inclusions using our new set of monoclonal antibodies in both US and Japanese Kawasaki disease patients. These antibodies did not bind to control lung, and our control monoclonal antibodies that we made did not bind to the KD lung. And I just want to mention that these antibodies that were very strong binders are highly genetically diverse. So we're not proposing that there's a skewing of a particular type of VH family member in Kawasaki disease. These are very diverse. They have variable CDR3 links, variable number of mutations, and they have different light chains. So now we're back sort of where we were before, except we know that we have the right light and heavy chain partners, and hopefully can now go towards identifying what the target of these antibodies is. And this is where pepperprint came in, and the arrays provided by pepperprint have proven to be extremely useful in our studies. So we had the pepperprint scientists prepare us a custom peptide array because we were strongly considering a Kawasaki disease to be likely to be due to a viral agent. We decided to have the array made from all the reported B-cell epitopes of human and animal viruses in the immune epitope database. And so this array was prepared. It includes peptides from a large number of RNA and DNA viruses. And I'm going to first show you the result of screening monoclonal antibody KD42H4, which is one of our strong binders on this array. When this antibody was applied to this array, multiple very similar peptides were shown to bind, and a motif was identified with a high level of significance. We then had the scientists at Pepperprint prepare us a substitution array where we used one of the peptides that was strongly binding to our antibody and replaced each position with all of the amino acids and determined whether there were specific amino acids that were absolutely required for binding and whether replacement of certain amino acids led to improved binding. And from this data, we made an optimized peptide for the binding to this particular antibody. We then said, well, if this is really a Kawasaki disease antibody, more than one patient should be making an antibody to this protein. So to test that, and our original antibody that I've described was from the fourth KD patient that we did our plasmablast analysis from. And so we tested all of our antibodies and found that we had additional antibodies that were also binding to this peptide. And these were from 3 different Kawasaki disease patients, and all of these children had developed coronary aneurysms. I should mention that there was another antibody from KD patient 6 that also showed very strong binding. So we decided we would want to determine whether it was binding to the exact same epitope of the peptide or maybe a slightly different epitope of the peptide. So we also performed substitution matrix analysis on this antibody using our optimized KD peptide. And basically, although I don't have these right next to each other, basically shows that the epitope is really the same. And again, I want to mention and emphasize that these antibodies that are binding to the same peptide epitope are genetically diverse with different light chains and different heavy chains. And this isn't just a clonal expansion of one particular type of antibody. So to further demonstrate that this protein sequence was present in the inclusion bodies in the KD patients, we took these 2 antibodies, we pre-incubated the antibody with a scrambled version of the peptide where we just put the amino acids in a different order, and we demonstrated that we still had very good binding. But when we took the specific KD peptide and pre-incubated the antibodies with that, we totally took away the binding. And this demonstrates that these inclusion bodies contain a protein that has this epitope. So to summarize this part, we made monoclonal antibodies that identify these inclusion bodies from 9 of 11 KD children, and 5 of 32 antibodies that identify the inclusion bodies recognize one specific peptide. Our other KD monoclonal antibodies that bind to the inclusions likely recognize other epitopes of this putative viral agent. And the blocking experiments demonstrate that there was a peptide epitope in the inclusion bodies. So in this next part, more quickly, I want to just mention that, you know, you could say, all right, this is a very kind of complicated way of identifying a serologic response to a peptide or protein antigen in Kawasaki disease. And if it's true, then you shouldn't have to get the plasmablast. You should be able to just take sera from KD patients, and they should recognize this antigen. So we developed a Western blot assay, and we prepared a KD peptide with 3 copies linked by spacers and expressed it as a fusion protein to get it into the gel so that we could perform a Western. And what we learned was that if we took KD sera after day 8 or so of illness, we could find significant recognition of this peptide. And for example, so this was a GST fusion protein we made, and in each of these lanes we have GST as the first lane, just to make sure that we're not having non-specific reaction to the GST fusion protein. And then this is the construct that has the 3 copies of the peptide in it. And then we just ran some human IgG as a positive control for our secondary antibody. So here's KD patient on day 11, on day 8, on day 28 in the pre-IVIG era, so never received IVIG, and these all had strong reactivity with the peptide. We didn't have sera from too many of our plasmablast patients prior to the day that we got their plasmablast, but we did have sera from day 3 of illness from patient KD4, and this patient had come to the emergency room a couple times before presenting with classic Kawasaki disease. So we were able to scavenge this serum. And you could see that on day 3 of illness, this patient whose plasmablasts recognize this antigen from day 8 is negative. But if we took sera from earlier in the illness on our other KD patients, they were generally negative. And the febrile control children are shown here. They're not reacting, and some infant controls as well. And we checked that we had good loading of our proteins. This is just a kind of a complicated slide. I don't want to spend too much time. I just want to emphasize that we had 5 out of 8 Kawasaki patients after day 8 that were positive by this assay from a number of different years. And as I mentioned, KD4 was negative on day 3. And then earlier in the illness, largely negative. And then from the febrile controls, mostly negative, although 1 positive. As you might expect in a ubiquitous agent, you might just happen to have that happen. So this just summarized the results of this part. We had 5 of 8 KD patients after day 8 that were recognizing this antigen, 1 of 9 febrile controls, and infrequent earlier in the illness. And these results are consistent with a ubiquitous causative agent. They'd be what you'd expect for a ubiquitous causative agent. So our future directions are to identify the genome containing this peptide sequence, and we have a very large database of high-throughput RNA sequence data from KD tissues. So we're searching for the translated protein epitopes in this data set. We also want to pursue additional antibodies that bind the ICI but don't recognize this first peptide, and we're in the process of doing additional experiments with the pepper print array assays. And then we would like to work some more on our serologic assays. We have some things in the pipeline to improve the assay, and we're working on all of that. So I want to make a little plug for why this is a great thing to do, not just for Kawasaki disease. So this tool, as I mentioned, has been used in the study of a number of different infectious diseases like dengue and HIV and influenza. And the power is that you can identify specific antigenic targets of B-cell response to known infections and in theory to unknown inflammatory diseases of unknown etiology. It's also a powerful tool for deciding what kinds of antigens the immune system is particularly targeting for the immune response, and you could apply this information to optimize vaccination strategies. So in summary, we have a child with Kawasaki disease with an unknown etiologic trigger. We identified antigen-specific plasmablasts, made monoclonal antibodies corresponding to those plasmablasts, showed that they bind to the inclusion bodies in the tissues. We used the pepperprint viral peptide array and substitution array to identify a peptide epitope, which we've shown blocks binding to the inclusion so that the epitope is present in these inclusion bodies. And then we've also demonstrated that patient sera also recognize this peptide. So this has been a group effort for sure. And I've been working on Kawasaki disease pathogenesis for decades. I don't have all my former lab members on this slide, but all of them have contributed in some way to this progress. The individuals who've particularly been involved in this work that I'm presenting today I've depicted on this slide, in addition to the collaborators that I have listed here. And I won't go through them all in the interest of time, but all of these individuals have played a key role in our success to date. So I think that's my last slide, and thank you for your attention, and I'm happy to answer any questions.
Great. Thank you very much, Anne, for this thorough and fascinating work that you presented. So I'm going to open up the floor for questions. So once again, if you do have questions, use the question box and type your query. So I do have a question here by an attendee. I think, Anne, this is the attendee is referring to one of the earlier slides in the presentation, and the question is 75% an upper estimate for plasmablast specificity? Those I know, and again, this refers to the person who asked the question, who have used plasmablasts for reverse vaccinology have had much lower success rates for antigen specificity, and that is in the low single percent digits. Can you comment about this, Anne?
So there, I don't know, I don't think I listed all the papers. There are quite a few papers that discuss that the general principle is that after infection, about 70% or so, 75% of plasmablasts are antigen specific in that limited time frame. I suppose the person asking the question might know of some exception, but these are some of the references, and these authors have pretty much agreed that this is it's true for vaccination as well as for natural infection. And there are some studies ongoing for autoimmune disease as well, if you can get a patient sort of shortly after they've start developing symptoms. So I can't comment on the specific question of it's true that the plasmablasts are only about 1% of the total B cell pool a lot of the time, but then when you take that 1% and you look at its antigen specificity shortly after vaccination or natural infection, that should be very, very much directed at the vaccine or the pathogen. Not necessarily all the same antigen, of course, because it depends on what's in the vaccine, or if it's a natural infection, there may be multiple different proteins of the agent that are being targeted.
Great, thanks, Anne. So here's another question. What is the life of a plasmablast? Would you know the life memory? Hold on. Would you know the life memory B cells? Sorry, a little less related to your presentation. So again, about the life of the plasmablast and whether you know the life memory of B cells.
So that's actually a really good question, and it's actually pertinent to what I've presented, because plasmablasts are very kind of sort of delicate cells, and it is important to process them and use them right away. I have waited up to 24 hours after obtaining the cells to do the flow, but I should say you can keep the blood overnight before you isolate the plasmablasts and do the flow, but not longer than that, and definitely not freezing the peripheral blood mononuclear cell pool and trying to pull out the plasmablasts later. That does not seem to work. They pretty much get disrupted by that.
Okay, great. So here's another question. So in a chronic disease with chronic stimulation, could the plasmablast be used to identify antigens?
That's a very good question. I think that there hasn't been enough work on that to know for sure. I think it could depend on whether there is evidence that the process is in good part B cell driven. But if there's evidence that that's the case, I think it would be worthwhile to try to see if there's clonal expansions in the plasmablast pool that might lead you toward deciding it was worthwhile to prepare antibodies and determine what the targets were.
Okay, thank you, Anne. So here's another question. Have you determined at what day of KD illness is the maximum height of detectable plasmablast levels? Is it dependent on severity of the KD illness?
That's another great question, and I have to say I don't know the answer to that one. We didn't, this technology, as you can imagine, is a little bit labor intensive, and we did not do the studies on patients presenting on each of the possible days of illness. In general, I would say our results appeared to be best if we stayed within the 1 to 2 week time frame. We did test a couple patients that were after the 14th day, and those seemed to have less clonal expansions, and I think things had settled back down to a level where it was more difficult to pull out the antigen-specific plasmablasts. But as far as the plasmablast numbers, we didn't focus on that because that wasn't the goal of our study. So I can't really answer that question.
Okay, thank you, Anne. So let's do 2 more questions. Here's another one. Can you elaborate a bit on the step concerning isolation of the monoclonal antibodies once the plasmablasts are isolated?
How do we make the monoclonal antibodies must be the question.
Yeah, that's, I would understand that.
Yeah. So we do the reverse transcription PCR on each well, each well contains 1 cell. So we amplify the variable region of the heavy chain and light chain genes. Then we clone those sequences into immunoglobulin expression vectors and prepare the antibodies by transfection in tissue culture cells. And this work is not our original work. So there are a number of papers in the literature which I can provide that discuss the actual technology. We made modifications to the transfection part of the protocol, and there are many different systems one could use to prepare the antibodies in tissue culture. But our paper is in Journal of Infectious Diseases. There's the preprint is available, and all of our detailed methods are there in the paper if the questioner would like to reference that.
Great. Thanks, Anne. So last question. And by the way, I invite also our panelists maybe to comment on this. Why did you choose to characterize isolated antibodies from single plasmablasts instead of screening the whole IgA serum profile? Or did you test this as well?
We did not submit serum for application to one of these arrays. I think that is one approach one could try. My concern would be that there would be a lot of non-relevant IgA in the peripheral blood, and I think that the advantage of our approach is that we can prepare a fairly large quantity of a specific antibody that could be applied to the array. So I think an individual IgA in the blood that might be relevant could be present in a low enough quantity that you'd have less success with the array and perhaps more background as well, because if you apply it to a pan-viral array and there's some IgA in there to some common infectious agents, you're going to have some background reactivity. So that's why we focused on making specific antibodies and using the monoclonal antibodies to screen the arrays. We hoped to find a cross-reactive, and we believe that's what we've identified, some sort of epitope that has some similarity to a known virus, but isn't that specific virus. And to do that, we thought we probably needed to have a fairly high concentration of antibody to apply to the arrays.
Great. Thank you, Anne. So thank you all for attending this webinar presentation, and we also want to thank our speaker. We will make the presentation slides and the webinar file available shortly. So once again, if you do have any questions further on, please contact the PepperPrint team directly. And also please check our website for updates, future events, or webinar announcements. Again, once again, thank you to everyone for dialing in. I will end this webinar now, and please check our website for future events. We look forward to having you again. Thank you.
Thank you.